Hello, I'm Maya Burton, and my final presentation concerns brain cell death in rats of different age groups after an incident of trauma. So, my research question is when both young rats of about 2 months of age and older rats that are fully neurologically matured both undergo an incident of brain trauma after 1 month, which will encounter less neurological damage, which is measured in brain cell death. We can connect both our animal behavior and neuroanatomy modules to the foundation of this experiment. The animal behavior module, where we investigated the behavior of fruit flies after experiencing the treatment variable of ethanol exposure, is similar in that we're investigating the behavior of rats after trauma and documenting the progress after a scheduled amount of time. Our neuroanatomy module is also helpful in that it provided methods in which we can analyze the brain and compare and contrast its parts, as well as providing guidance on myelin and Nissl staining, which is particularly helpful since this experiment also involves brain staining. So I want to use that foundation to look into my research question. I found I wanted to look at what occurs in the brain of a rodent when there is any sort of trauma to the brain, and what primarily happens is called apoptosis or cell death. However, I had to be careful in deciding what age group I wanted to look at because there is a natural age range in rats where cell death naturally occurs in the brain, just because that's how the brain develops. And that age range is about up to 10 days postnatally. So as long as I looked at young rats past 10 days, that shouldn't have been a variable that interfered. Lastly, I had to decide how long after the incident of brain trauma I would look for changes in the brain. In a similar brain study conducted with 3-month-old rats, brain cell loss was looked at 1 hour, 2 hours, 48 hours, 1 week, 2 weeks, multiple months, and then a year after traumatic brain injury, and damage was seen over a year later. So, while using both old and young rats, I thought 1 month was an appropriate amount of time to gauge which age group suffered the most cell death in the brain, based on Smith et. So, going into my experimental design, my official hypothesis is if a rat of about 2 months of age and a neurologically matured rat of a year old both undergo traumatic brain injury, the younger rat will show more brain damage after a month than the mature rat. The treatment variable is their age. It's the only thing that differs between the two rat groups, and the matured rat will be used as a control. The constants include the tunnel method, being used to observe cell apoptosis, the anesthesia they're put under, the method of traumatic brain injury, and the duration of time they're looked at after the injury. Randomization occurs through the order in which they're taken through the experiment so that there's no experimenter bias in simulating the brain injury. And there's replication because there's 15 rats used for each. My procedure is based on the same foundation as the study done by Rink et al., done on 3-month-old rats. So first, to standardize the traumatic brain injury, both rat groups will undergo anesthesia injections of sodium pentobarbital and hooked up to fluid percussion devices, which are the devices that actually produce the brain trauma by injecting saline rapidly into the brain. This method of TBI, or traumatic brain injury, is very similar to a human brain injury. We take 6 micrometer pieces of coronal slices from the mid-hippocampus region. Because I'm assessing damage 1 month after the injury, these slices are taken a month after the simulated head injury. I'd store them using paraffin blocks, which are just boxes you can use to store samples for a long amount of time. And the easiest way to assess the neurons in the brain seems to be with what's called tunnel histochemistry, where terminal deoxynucleotide transferase, deoxyuridine triphosphate, or DUTP nick end labeling, all abbreviated as tunnel, detects apoptotic DNA fragmentation in the brain. The staining will be completed. We deparaffinize the samples and rehydrate them for viewing, similar to what we did with myelin and Nissl staining. And that's when we can look at the brain using electron microscopy. In my results, I predict that we'd be able to see more cell death in various regions of the brain in the mature rat, and a large part of this prediction comes from the study previously mentioned by Rink et al., where these brain areas showed the most damage after TBI. The areas of the brain I would expect to see more cell death in is the cortex, the hippocampus, a more concentrated area of the hippocampus called the dentate gyrus, and in the white matter of the coronal brain sections. This first visual is a model of where we'd be looking at apoptosis in the brain. The second visual is a more detailed version of where we'd be looking and where we'd find neuronal cell death, so the cortex, hippocampus, white matter, and dentate gyrus. Okay, in the results, we see the areas where apoptosis would occur for both the young and old rat, but the reason why young brains would encounter more substantial consequences is that the brain is still developing to an extent. Developing neurons encounter this damage, and although the brain has great plasticity, this trauma is part of the brain. This hypothesis is supported by studies like the one conducted by Dang Bryant at Alia, where ketogenic diets prevented alterations in young rat brains after trauma, but not in old rat brains. And this leads me to believe that young rat brains are more susceptible to environmental change than adult rat brains. So there are a few reasons why studies like these are helpful in the grand scheme of things, including providing research on the human brain's ability to bounce back after physical trauma. Considering brain trauma is the leading cause of death in children, it's particularly important to be aware of how TBI affects younger brains differently, especially in areas where brain injury is so pervasive, like maybe in sports. Neuroscientists would use this to study to answer questions about the long-term effects of brain damage, how brain damage affects different age groups, and what happens after incidents of brain trauma. And here's a list of my references.